Hello everyone, welcome back to one more video session and welcome to YouTube. Here I have come with another interesting topic, that is blue-white screening, taken from the chapter biotechnology or genetic engineering. Here screening indicates selection procedure, selection of transformants over the non-transformants. That means you need to select a recombinant, that means a host organism which is used in genetic engineering has carrying the recombinant DNA, or non-recombinant in the sense like the host which is used is not carrying the recombinant DNA. That's it. So how are you going to differentiate, that is transformant over the non-transformant? For that we are using this type of screening, blue-white screening, or there are other screening methodologies also. You can also go with antibiotic screening methodology, where I'm not explaining that, but I can give you the brief idea about it. To make you understand the concept, let me go with a small bacterial cell. We all know that it contains its genomic DNA and it has got autonomously replicating extra chromosomal circular DNA, that is called as plasmid. So this is the plasmid, which is double stranded DNA. Why plasmids are normally present in bacteria? In our case, we have immune system which actually protect us from the foreign particles or the pathogens or even antigens. But these bacteria doesn't have that immune system. Instead of that, it has got a plasmid which normally has genes in it. These genes could be producing certain resistance type of proteins which actually neutralize any type of antibiotic. For example, if I take this bacteria and put it in a Petri plate containing an antibiotic, we all know that antibiotic is a chemical which actually suppress or retard the or inhibit the growth of the bacteria. So I'm taking antibiotic which is called as ampicillin. I write amp here, where this particular plasmid has ampicillin resistance gene which could suppress or neutralize the antibiotic, that is ampicillin, so that the bacteria survives. What if this particular the gene is absent? In this case, ampicillin resistance gene is absent. That means it cannot survive. That's it. So I can actually exploit this property of the plasmid for genetic engineering. Similarly, it can also have another gene which could produce another product, that is, for example, beta-gal gene, which produces an enzyme called as beta-galactosidase. For example, instead of ampicillin, if I take a chromogenic substrate, that is X-gal, and if I grow this bacteria inside this X-gal, what will happen? This beta-gal gene produces beta-galactosidase, which reacts with X-gal and gives us the kind of color that is blue. That means the bacteria still has this beta-gal gene, yes, which gives us blue color indication. So I'm using this now to explain blue-white screening. And now, what is exactly this construction of the rDNA and what is transformation? Let me explain that. In human females, in the mammary glands, in the breast, there is specialized cells called as epithelial cells. Epithelial cells. If I isolate that cell and the DNA containing a specific gene which produces a type of protein, which is, this is the DNA, and which produces a type of protein called as alpha lactalbumin, which is very, very important for the growth and development of the infants. What if, like, we remove this gene, isolate the gene from the DNA, and then actually ligate it to some vector DNA like plasmid and make rDNA and put it in the host like bacteria like E. coli and multiply, so that we get lot of alpha lactalbumin, right, through downstream processing. So we can actually commercially exploit this. So this is how it is. So it helps so many poor kids. So they also get this alpha lactalbumin for their development and growth. Similarly, what I'm going to do is I'm going to draw this plasmid so that you can see it. This is the plasmid now. Now I'm taking only one particular gene to explain the concept, that is, this is beta-gal gene which produces the enzyme beta-galactosidase, and I'm using a specific restriction enzyme, because in genetic engineering we need to use restriction enzymes because they recognize the palindromic sequence, that is called as recognition site or restriction site, which is going to bind to the restriction site and cleave it. Cleave it means cut it. For example, PST1 is a restriction enzyme which is taken from a specific bacteria, that is Providentia stewarti. a gram negative bacillus, okay, bacteria. So this PST acts on this particular segment of the beta gall somewhere else here, and cuts it. That means this particular DNA segment has come out. That means beta gall gene is insertionally inactivated. Okay, still I have not inserted alpha lactalbumin, but I have inactivated it by action of PST1 restriction enzyme. Okay, I have removed this piece now. So that means we understood that beta gall gene cannot now synthesize beta gal or galactosidase enzyme. Enzyme cannot be synthesized. Done. Now what I do is I take this alpha lactalbumin and now gene and now ligate it here. So this is a plasmid. This is a plasmid. Now it is called as vector. And now after adding this alpha lactalbumin, I cannot call this as vector. No, earlier it was vector. I forgot to mention it. But now after adding this, now this DNA is called as rDNA, recombinant or recombination DNA. Why? Because it is having a part of around 70% of it is the vector DNA and 30% is gene of interest. So in this case, alpha lactalbumin acts as our gene of interest, or also called as foreign DNA. And one more thing I forgot actually, normally plasmid, why we prefer plasmid in this genetic engineering? Because plasmids normally carry certain properties, like it has got selectable marker genes. For example, ampicillin resistant gene is a selectable marker. Beta gall gene or kanamycin, tetracycline, chloramphenicol are the selectable marker genes. That's it. Okay. So here beta gall gene is our selectable marker gene. I have inserted my gene of interest into it, and now I have constructed rDNA. Next step is I'm going to introduce this into the host organism. So this is the host. Normally we prefer E. coli bacteria because it is easily available and easy to grow in any culture medium, and also their multiplication time is hardly 20 minutes. So within 20 minutes I get 2 bacteria. That means if I introduce this by a mechanism called as, or a process called as transformation, this methodology is called as transformation. I'm just putting this rDNA into the host cell. Okay, so let me just draw this here. So white color indicates our foreign gene ligated to the vector, yellow color gene. Okay, fine. So this is now transformed cell. As all these steps are happening in the molecular level, you cannot see through your naked eye. How do you know that this particular E. coli has taken rDNA? That's it. For that, you have to use screening methodology. Very simple. I forgot to mention one more thing. During transformation, we have to use different methodologies. For example, it's called as competency. The different competency methodologies are: if it is bacteria, is the host organism, then go with the heat shock treatment. If it is plant cell you are using as host, then go with biolistics or gene gun method, that is gold and tungsten, coat with the DNA. And then if you are using animal cell, then go with the micro injection. So competency, I'm just going with the heat shock treatment, and this particular host got the rDNA. And now I'm going to select it. Okay, I'm going to select it in a procedure where I'm going to grow this particular E. coli, that is now transformed cell for me, because I know it is transformed now, and in a chromogenic substrate called as X-gall. And what's going to happen? We know that this was the segment where beta gall gene was there, but we have insertionally inactivated by introducing our gene of interest into it. So now there is no beta gall gene, and now the beta gall enzyme cannot be synthesized, and you are not going to see any color here. So you see only white color. The colonies, this bacteria now multiplies, and it's going to multiply into several million folds, and in the Petri plate you see only white colonies. That means if you see white colony in the chromogenic substrate X-gall, then obviously your experiment, whatever you have done so far, is successful one. That means the gene of interest is now going to produce alpha lactalbumin, and then you can extract this from the bacteria through downstream processing and commercialize it. What if here in the step number 1 itself we have failed to ligate our alpha lactalbumin gene of interest? We failed to inactivate this beta gall gene. That means it is still having the beta gall gene, which produces beta gall enzyme, which reacts with the X-gall chromogenic substrate and gives you the blue color colonies. See, If you see blue color colonies, means whatever the experiment I have done so far is just a failure. That means this is a non-transformed cell. This is a non-transformed cell. If it is carrying the gene of interest, that means it is a transformed cell. That's how we use blue-white screening. Blue colonies indicates non-transformed cell, white colonies indicates transformed cell. That means your experiment is a successful one. Hope you understood the concept. Thank you very much.